Good morning, students. Today we will learn about the cells, human cells, and structure and the specific functions of each of these organelles or structure of cell. Now, what is a cell? Cell is a structural and functional unit of any living organism. Structural because with the composition of billions of cells, a particular structure, whether it's an organ or organelle, is made up. And functional because the function of the single cell by which the organ is made up and the function of the whole organ is same. So in definition of cell, you must have to mention the both terms structural and functional unit. Now, as per the organization of cell structure, cells can be of various types like prokaryotes, eukaryotes, and others as well as structures. And we human beings contain a eukaryotic type of cell, that means with a proper nucleus and proper organization of cell structures. And this is a diagram showing the composition of cell is mainly bounded by a limiting membrane, that is that cell membrane, and the rest of the portion of cell inside the cell membrane is called protoplasm. Now this protoplasm is again divided into two portions. The center portion of protoplasm is called nucleus, and the rest of the protoplasm except for the nucleus is called cytoplasm, which includes various types of inclusions and organelles, and some of these organelles are membrane bound and they are called membranous organelles like endoplasmic reticulum, Golgi apparatus, mitochondria, lysosome, peroxisome, and some of them do not contain any membrane, so they are non-membranous like ribosome, centrosome, cytoskeleton, etc. And here is the parts of the nucleus. As you can see, now this is a picture showing the structure of cell membrane as we can see it through the light microscope. As basically this portion is the cytoplasm, and in center there is nucleus, and in the center of the center, that means in the center of the nucleus, there is also another nucleolus, we call it. It contains chromatin, the nucleoplasm, and this is the limiting membrane of nucleus, nuclear membrane. Now this is a more elaborate picture showing the different types of cell organelles, cytoplasmic organelles. As you can see that this is a nucleus, and in close relation to nucleus there is endoplasmic reticulum. Some of the endoplasmic reticulum contain ribosome, which is a granular structure in the surface, and because of presence of this ribosome, this part of endoplasmic reticulum is called rough endoplasmic reticulum. And some part of endoplasmic reticulum do not contain any ribosomes, so this part is called smooth endoplasmic reticulum. And there is also Golgi apparatus. These are the Golgi apparatus, the vesicular and tubular structures, and these are the mitochondria. Then there is lysosome and cytoskeleton, microtubules. Now this is 3D animation showing the structures of cell. This is a hemisection of human cell. Inside this will come to the elaboration of the function of the particular structures of cell. This is the cell membrane. Cell membrane, will know that it's a thin structure that is elastic, that can adjust its shape. It envelops the whole cell, and it's called cell membrane. Its thickness is about 7.5 to 10 nanometer, and it is composed of protein, mainly lipid, and also other carbohydrate like substances. And on the basis of how much piercing the protein has done to the cell membrane, it's called integral protein. If it pierces the cell membrane wholly, it's called integral protein. That means it's the integral portion of the cell membrane. And if it is attached only to outside or inside, that means it at the one side of the periphery of the cell membrane, then it will be called peripheral protein. And in lipid, there are various types of lipid, as you can see. This is a fluid mosaic model structure showing this is the most acceptable structure of cell membrane by far. According to this model, cell membrane is a lipid bilayer structure which contains also protein and carbohydrate on the surface, and it's a semi-permeable structure. We can see here these are the integral protein because it has pierced the cell membrane wholly, through and through. So that's why they are called integral protein. But in case of peripheral protein, you can see that they are attached to integral protein either inside, or they may also attach to outside of that means at the periphery of integral protein. And these are the carbohydrate attached to the lipid. Carbohydrate, if it is attached to lipid, then it will be called glycolipid. Glyco means carbohydrate. If it is attached to lipid, then in that case it will be called glycolipid. If it is attached to protein, in that case it will be considered as glycoprotein. Okay, now come the functions to the protein. The integral protein, the protein that has pierced the cell membrane through and through, they provide a tremendous number of functions. As we all know that our cell membrane is a semi-permeable structure. That means that it will not allow any type of substance to pass through it easily. So that's why semi-permeable. So how if the cell membrane is lipid soluble, so it's understandable that it will only allow the lipid soluble substance to pass easily through it. So there must be a mean by which the water and water soluble substances like oxygen and other necessary nutrients can enter into our cells, because they are very much necessary for the cell to survive. So these needs are or these means are provided by the protein. They serve as a channel by which the water and water soluble substance can easily pass. They can also act as a carrier protein to carry the various important substances, and they may also act as receptors for the various water soluble substances like hormones, especially for peptide hormones. And they can also act as enzyme and enhance the rate of various types of metabolic reactions. And in peripheral protein, in case of peripheral protein, they almost entirely act as enzyme, but sometimes they may also control the types of substances that will pass through the cell membrane pores. Now comes to the functions of lipid. The main function of lipids, especially the phospholipid, is to maintain the permeability of cell, allowing only lipid soluble substances to pass through the membrane. Okay, and sphingolipid protects cell from harmful environmental factors, helps in signal transmission, acts as adhesion site, and cholesterol helps to determine the degree of permeability and control much of the fluidity of membrane as well. Now there may a question that may arise in your mind, that why this maintenance of semi-permeability is so important. Why it is permeable to water or water soluble substances easily? What will happen? Because this will actually create a havoc in our cell, because if water can pass easily inside and outside of the cell, then This particular function of cholesterol, I would like to mention it here. The degree of fluidity of cell membrane will not be maintained. Sometimes it will be too swelled that it can burst, and sometimes it can become shrinkage. That means it can become dry or it can shrink. So in that both condition, our it is not you know good for our body. So for this particular reason, maintenance of the permeability of the cell and to make it semi permeable, that means only the lipid soluble substances can pass. Water soluble substances can also pass, but not so easily. If they have to pass, they have to pass through a certain means. Now comes the functions of carbohydrate. The carbohydrates, if sometimes this question may arise in your mind, that why our cells don't get piled up, why they do not attach to one another, and why they how they keep separated from each other. So this means of separation is provided by carbohydrate, because carbohydrate on the surface of each cell in form of glycoprotein, they provide a negative surface charge in each of the cell, and as we all know that the same charge distracts one another and the opposite charge attracts one another. So if all of the cell contain glycoprotein on their surface, so all of them have negative surface charge, so it's natural that they will distract one another, so it's normal for them to be keep separated. Otherwise they will pile up and form a relux, which is found in various pathological conditions. Now, in another form, in the form of glycocalyx, when it's needed, then carbohydrate can also provide the means by which the cell can attach themselves when it's needed. Okay, and many of them can also act as receptors for binding hormones, and some of them also help in immune reactions. As we all know that some of us have blood group A positive, or some have B positive, B negative, O negative. Why does this happen? Because we all have different types of blood antigens on our cell surface, and these blood antigens which are present in the RBC or red blood cell surface, these are actually glycoproteins. So that means carbohydrate also play a role in this. And if you there is mismatched blood transfusion reaction, that means if you transfuse a positive person with B positive blood, then there will be vedio incompatibility, and so it's immune reaction, and carbohydrate is playing a role in this immune reaction. So this is the meaning of this line. Now comes the structure and functions of cytoplasmic organelles. We have covered the cell membrane. Now we are going inside of the cell membrane. What is inside in the cell membrane? So inside of the cell membrane, I have already mentioned that in the center we've got nucleus, and the rest of the portion is called cytoplasm. So first we will go for the cytoplasmic organelles. But first organelles would like to discuss is endoplasmic reticulum. I think I have already discussed it somehow in the diagram previously mentioned. Endoplasmic reticulum also, it is a membranous structure, so definitely it contains an endoplasmic membrane, and inside the membrane there is endoplasmic matrix space, and this is a diagram showing endoplasmic reticulum. Now come to the functions of endoplasmic reticulum. As the granular endoplasmic reticulum or rough endoplasmic reticulum contain ribosome on their surface, so functions of ribosome is to synthesize protein, and so function of rough endoplasmic reticulum will be also to synthesize protein. Very easy to understand. And the smooth endoplasmic or granular endoplasmic reticulum, they do not contain any ribosome on the surface, so they cannot synthesize protein, but they have also other important functions like synthesis of lipid substances. Only protein is not necessary for our body, but also lipid. So this lipid is synthesized by smooth endoplasmic reticulum, and various types of toxin in our food or drugs that we consume entering into our body each day in every minute. So this the substances also contains some part of it that is not needed for our body. Rather than if it is stored in our body, it can act as a toxin. So this toxin must be go through a detoxification process in which they will not further remain harmful for our body. So this detoxification process is done by the smooth endoplasmic reticulum, and also they are important for providing enzyme for glycogen breakdown to provide glucose from glycogen. As we all know that glucose is necessary for our energy, and the glycogen is the storage form of glucose. When we consume too much of glucose which is not immediately required, our body stores the glycogen in liver, specifically in liver, and then when it is required, then they break down this glycogen and deliver it to the desired site by in the form of glucose. Now comes to the mitochondria. Mitochondria is called powerhouse of the cell. Why powerhouse? Because it's providing power or energy to our body. How? Because our body cannot utilize glucose, protein, or lipid, etc. They can only utilize ATP. Anything we consume, anything that is either a carbohydrate food or a protein food or lipid food, it must have to be converted through a metabolic series of reactions to adenosine triphosphate. In only that form the body can utilize this food. So this is the primary source of energy, and is this ATP is produced in the mitochondria by the means of oxidative phosphorylation. That's why mitochondria is called the powerhouse of the body. So mitochondria also, as its membranous structure, must will contain a mitochondrial membrane, and this membrane has a speciality that this inner part of this membrane is folded inward to form a shelf-like structure which we call cristae. This cristae is very much important because they provide a surface area for these metabolic reactions to occur, and they also act as a source of enzymes that are needed for these metabolic reactions. Inside the mitochondrial matrix, the rest of the portions, except for the mitochondrial membrane, the inside portion is called matrix. This matrix also contain enzymes for a number of metabolic reactions. We can see in this no uh, functions. Cristae has oxidative enzyme that is needed for oxidative phosphorylation. This is the very reaction by which the adenosine triphosphate or ATP is produced. And mitochondrial matrix also contains enzyme necessary for oxidation of nutrients, specifically the beta oxidation of fat, TCA cycle, etc. And this topic has already been talked, and cristae also provide a large surface area for this chemical reaction to take place. Now coming to the another very important membranous structure that is called the Golgi apparatus. Golgi apparatus is a very simple structure containing stalk layers of thin vesicles or enclosed vesicle lying near one side or other side of the nucleus. So what is the function of this Golgi apparatus? The function of Golgi apparatus, as we have previously seen seen that the RAF endoplasmic reticulum function was to synthesize protein. So when they synthesize protein in their ribosome, these protein are in a raw state. That means some protein are very large, some are very small, some contain the unnecessary portions that are not needed for our body, and some do not contain some necessary portions that are required by our body. So they must have to go through a process of modification before they are delivered to the destination. So this particular modification or processing of this protein, RAF protein made by the RAF endoplasmic reticulum has taken place by the Golgi apparatus. So in a nutshell, if we can summarize the functions of Golgi apparatus, is to synthesis of protein, packaging of protein, processing of protein, and delivering of protein. I'm sorry, synthesis of protein is not a function of Golgi apparatus, it's a slip of tongue. So in one sentence, if we can summarize the function of Golgi apparatus, it is to packaging, processing and delivering protein. They first will process the protein or modify the protein, then they will put it into a bag of membrane that is called the vesicle. So in this vesicle they will put the protein and they will deliver this vesicle to the desired site where it is required. So packaging, processing and delivering, this is the functions of Golgi apparatus in a nutshell. Now comes to the lysosome. Lysosomes are vesicular organelles which are which have made from the Golgi apparatus, and lysosome function is to here is a picture showing the lysosome, and you can see inside the lysosome there are number of enzymes present in it, and the function of ribosome is to actually hydrolyze the hydrolyze means what means it hydrolyzation or hydrolysis means to break down anything with the help of water. Okay, so this particular reaction is enhanced by the hydrolase enzyme that is provided by the lysosome. Now the question arises that what type of things or substances can go through hydrolysis by lysosome. Lysosome can actually hydrolyze anything, okay, but it particularly hydrolyze the food particles that we ingest, that is ingested by the cell. uh, if it's not digested, then we cannot consume it or we cannot utilize these food particles, and unwanted matters like anything that is foreign to our body, anything that with which our body is not acquainted with, will be digested by lysosome because it will consider it as an enemy, so it will provide the digestive enzyme, and in this means it the foreign body like if it's bacteria or virus or anything, it will be destroyed. And also damaged cellular structure is also digested by lysosome. Now this may raise a question in your mind that why damaged cellular structures. Food particles we can understand, if they are not hydrolyzed we cannot consume energy, and unwanted matter like bacteria, protozoa, these are also understandable because if they are not hydrolyzed then they will be harmful for our body. But why damaged cellular structures? Because damaged cellular structures are also not needed for our body, and anything that is not needed by our body, whether it's a damaged cellular structure or a aged cellular structures, when a cell completes a certain type of life, I mean certain life expectancy, then it cannot further contribute the functions of body, and anything that cannot further contribute to the functions of our body is not needed by our body, and it has to be removed from the body, because if they piled up in the body, then the new cells or the new structures maybe get hampered. So that's why they must have to be removed from the body, and this particular function is provided by lysosome. Now comes the peroxisome. Peroxisome formed by the self-replication or budding of smooth endoplasmic reticulum, and their function is to help protecting the cell from poisonous substances by the means of oxidase enzyme. Now what is this poisonous substances? Poisonous substances doesn't necessarily means the poison or toxin, you know, because all of us are not going to commit suicide, you know. So poisonous substance here doesn't necessarily means poison or toxin. It means that every substances that we consume, even evenly or seemingly very innocent vegetables or meats or fish, anything, any food particle or drugs, you know, or in western countries they have the habit of consuming alcohol. So anything that you consume, it has a it has to go through a number of metabolic reactions first to be consumed or to be utilized to our body, and in these metabolic reactions when the necessary or the desired substances are produced. The unnecessary substances are also producing as a byproduct, and in these unnecessary substances, the most common is free radicals. As I have already mentioned, that ATP is the form by which our body can utilize energy, and this ATP is formed by oxidative phosphorylation. So it means oxygen must have to be present for this reaction to take place, and this oxygen is when oxygen is utilized in any reactions. In this reaction, ATP will be produced, but as a byproduct, free radicals will be also produced, and these free radicals are a product of oxygen that are not natural oxygen. They are either superoxygen or hydrogen peroxide or sulfur dioxide. These are not needed for our body, rather than they are harmful for our body, so they must have to be removed or detoxified from our body. So this detoxification is process is called peroxidation, and this peroxidation process needs peroxidase enzyme, and this enzyme is provided by the means of peroxisome. So this is the function of peroxisome. There's a common question that may ask in the what are the difference between the lysosome and peroxisome. So first difference is lysosome is made up from Golgi apparatus, and peroxisome uh, is a structure which is formed from the self-replication or from budding up of smooth and plasmic reticulum. Lysosome contain hydrolase enzyme, whereas peroxisome contains peroxidase enzyme. The functions of lysosome is to digest the food particles or unwanted matters or etc. In a nutshell, the function of lysosome is to digest. Okay, and the function of peroxisome is to protect what to protect ourselves from the harmful consequences of free radicals. Now, the non-membranous organelles. In non-membranous organelles, first important organelles is ribosome, and in ribosome, if they are attached to plasmic reticulum, they will be called attached ribosome, and they may also remain free in the cytoplasm. But whether they are free or attached to endoplasmic reticulum, their main function is to synthesize protein. In case of attached ribosome, this protein is transmembrane protein, and if they are free in the cytoplasm, then this protein is called cytoplasmic protein, that is synthesized by the free ribosome. And these are the pictures showing the cytoskeleton. Cytoskeleton is a network of fibrillary protein. They can be depending on the diameter, they can be regarded as microfilament, intermediate filament, or microtubule. Diameter is different in each of them. And the microfilament are basically actin and myosin filament, and they are present in muscles, and they helps to as a elastic supports to the cell, and they helps in muscle contraction. In the chapter of muscle contraction, that portion will be much elaborately discussed, so I am not going for it. Now comes the functions of microtubules. Various types of microtubules are present in our body in the forms of flagellum, cilia, spindles, in mitotic cells, centrioles, etc. What are the functions? First function is that they helps to provide a rigid physical structures for the cell. That means the cell structure, for it to be maintained, they have the need of microtubules, and they also determine the cell shape. Other Without the help of microtubules, the definitive type of cell shape maintenance will be not possible. And it also helps in forms of spindles, centrioles in the cell division. You all have passed through the steps of mitotic division, and so you can see the various steps like prophase, metaphase, anaphase, and in these phases you have already known about the spindles, centrioles, and these are actually the microtubules or cell cytoskeleton, and they also allows the cell to move. You can see that some cell contain contain flagellum, by which they can move from one place to another place, which is very much needed for our body. Like the most common example of this type of cell is sperm. For sperm to fertilize ovum, they must have to run, and they must have to go in a very rapid speed towards the ovum, and for this particular purpose they need their microtubules in the form of flagella. If the more than 20% of sperm in semen is non-motile, that means if more than 20% of the sperm doesn't contain flagellum, or if the flagellum cannot work properly, then this person will be infertile. So this is a very small structure, microtubules, but these microtubules can also play a very significant role in a person's life. So all of these cell organelles, or all of these cell structures actually are needed to be learned, because all of them play an important role in our life. And last of but not least, that they provide a track like system for movement of the organelles. If you can see, I would like to recall this slide. Recall this slide. Yeah, this is the cell cytoskeleton nanomotor containing the substances. So these are nanomotors are motors that we have gotten naturally from God. They helps to transport of various substances, and this cytoskeleton provide a way, or a system through which they can actually walk through this. Now the nuclear parts of nucleus have previously been discussed, so I'll not go for it. The main function of nucleus, as you all know, that nucleus is called center of our body. So why it's called center of our body? Because it contains the chromatin, and in chromatin it contains the genetic information in DNA. So this genetic information is the main blueprint by which it is determined that what type of cell it will produce, what type of function it will perform. So I mean, decision making is been taking place in nucleus. So in that sense, it is called the center of the cell, headquarter. Okay. This gene is in the DNA determine characteristics of cell protein, then controls cytoplasmic and nuclear activities. Gene also promote reproduction of cell RNA, nuclear required to synthesize protein. So all of these vital decisions are made up in the nucleus. So that's why nucleus is called the center of the cell. And these are the intercellular junction between two cells by which they can communicate with each other. Tight junction, desmosome, Juna adherence, hemidesmosome, focal adherence, and junctions that permit the passage of ions and molecules. These junctions, number one type of junctions, they do not help in transport of substances. They always just fasten the cells to one another, attach the cell to one another. The second type of junctions helps the passage of ions from one cell to another, like in gap junctions or tight junctions. In tight junctions, resistance is high. In gap junctions, resistance is low. That's why it's called gap junction. It's called tight junction. Intercellular functions, intercellular junctions functions are described in this slide. They have to attach cell to one another and passage of ion. You know. So this is all about cell. Thank you very much. If you have any question regarding the video, then you can ask me directly in the class, or you can comment on the video. Thank you very much.